How are oxygen and carbon dioxide transported within the respiratory system?
After gas exchange in the lungs, the body relies on efficient mechanisms to transport oxygen to the tissues and remove carbon dioxide, a metabolic waste product. Oxygen is carried in the blood primarily by hemoglobin, a protein found inside red blood cells or erythrocytes. Each hemoglobin molecule contains 4 iron-rich heme groups, and each can bind one oxygen molecule, allowing each hemoglobin molecule to carry up to 4 oxygen molecules at a time. When blood flows through the pulmonary capillaries, oxygen diffuses from the alveoli into red blood cells and quickly binds to hemoglobin, forming oxyhemoglobin. The partial pressure of oxygen, or PO2, in the lungs ensures efficient loading of hemoglobin. As oxygen-rich blood circulates to body tissues, local conditions such as lower PO2, increased PCO2, lower pH, and higher temperature promote hemoglobin to release oxygen where it is most needed, a phenomenon known as the Bohr effect. Clinically, the efficiency of this system is assessed by measuring the oxygen saturation with a pulse oximeter or by analyzing arterial blood gases. Hemoglobin's ability to carry oxygen is central to patient care. In anemia, reduced hemoglobin levels mean that even with normal oxygen saturation, total oxygen delivery can be dangerously low, resulting in fatigue, pallor, or shortness of breath. Abnormal hemoglobin or carbon monoxide poisoning can also severely impact transport. Carbon monoxide binds to the iron in hemoglobin with an affinity of about 200 to 250 times greater than oxygen. When carbon monoxide occupies these binding sites, hemoglobin can no longer carry oxygen effectively. In addition, carbon monoxide binding causes hemoglobin to hold on to any remaining oxygen more tightly, producing a left shift of the oxygen-hemoglobin dissociation curve, so even less oxygen is released to tissues. The result is severe tissue hypoxia despite normal or near normal blood oxygen levels. Now let's look at carbon dioxide transport. Carbon dioxide produced by cells during metabolism is transported from tissues back to the lungs in 3 main forms. The main form is as bicarbonate ions. About 70% of carbon dioxide is carried in this form. Inside red blood cells, carbon dioxide reacts with water to form carbonic acid, which quickly dissociates into bicarbonate and hydrogen ions. Bicarbonate ions diffuse into the plasma. This reaction, catalyzed by the enzyme carbonic anhydrase, is crucial for maintaining acid-base balance in the blood. The next way carbon dioxide is transported is bound to hemoglobin as carbaminohemoglobin. Around 20 to 25% of carbon dioxide binds to the protein part of hemoglobin, but at a different site than oxygen. The remaining 5 to 10% of carbon dioxide travels dissolved directly in the plasma. At the lungs, these processes reverse. Bicarbonate is converted back to carbon dioxide gas in red blood cells, and carbon dioxide is released from hemoglobin. The resulting carbon dioxide then diffuses from the blood into the alveoli and is exhaled. Blood gas analysis provides direct measurements of arterial oxygen, carbon dioxide, and blood pH, allowing clinicians to evaluate how well the respiratory system is delivering oxygen and removing carbon dioxide. The bicarbonate buffer system is a critical mechanism for regulating blood pH. Disruptions can result in respiratory or metabolic acidosis or alkalosis, each requiring rapid medical intervention.